Okay, I'm going to try to give the internet an update on what normal iron metabolism looks like in 2017. The first thing I want to clear up is what happens to iron once you swallow it. It's in your stomach. It's in an acidic environment. So what does the acid actually do? There are a few sources that say that the stomach acid reduces the iron from an Fe3 plus to an Fe2 plus state, and then almost every other source, several other sources say that that is definitely not the case, that the iron is maintained in a soluble state by the acidic environment, that this acidic environment actually changes it from an Fe2 to an Fe3 plus state. It oxidizes it. We're left with a paradox because we know that only Fe2 goes into the cells, and we know that whenever you take things like proton pump inhibitors or antacids, it reduces iron uptake. The answer to that is probably that the low pH allows the iron to be maintained soluble. It removes it away from proteins that would bind it up, and it also prevents it from condensing down and nucleating. So just really quick, since we're talking about the stomach, there are other things that you can eat that can prevent you from absorbing iron. Those things include tannins in your tea, and there are a few other foods as well. Ascorbic acid can help you absorb it. It was once thought that the mechanism of this was that it directly reduced the iron to Fe2+, and now it's thought that the ascorbic acid goes into the enterocyte and accepts the electron from the iron reductase. Iron gets absorbed from the duodenum of the GI tract, and this is the basic layout of the duodenal enterocyte. So what happens is the Fe3 gets reduced into Fe2 by something called duodenal specific cytochrome B, or D-Cyte B. This is an iron reductase. After it gets reduced to Fe2, it can go through the divalent metal transporter. After it's into the cell, the options are get stored in ferritin or get shipped out through ferroportin. Now there's a conundrum amongst the various textbooks and journal articles that I've read and the pictures that I've looked at as to exactly where, so once the iron comes into the cell, it comes in as Fe2, it gets converted back into Fe3 at some point. Does it stay there? How long is it there? Why is it in Fe3+? These are all questions that I have not found the answer to, but it seems like it gets converted into Fe3+ as it's stored in ferritin, because ferritin heavy chain has an iron oxidase activity. But every transporter that iron can move through requires the iron to be in an Fe2+ state. So it moves through a DMT1 transporter, it moves through a ferroportin transporter. Those all require it to be in an Fe2+ state. The major regulator for iron leaving the enterocyte is hepcidin. Hepcidin will bind to ferroportin. After it's bound, it will cause it to be degraded and endocytosed. There are a few other things that impact the enterocyte's ability to absorb iron and to create ferroportin and excrete it. Specifically, when oxygen status is good, the enterocyte's happy. It doesn't want iron, it doesn't need it, it's fine. Whenever you become hypoxic, maybe anemia for example, you start to express hypoxia-inducible factor. In fact, hypoxia-inducible factor is released and it can go into the DNA and transcribe. So it will bind to the hypoxia response element. In the in the duodenal cells, it will upregulate the production of DMT1. And it's easier to think of it as your body says, hey, I don't have any oxygen, so I must be low on hemoglobin, so therefore I must be low on iron, so I better take in some more. The liver's a little bit weird, so the liver will also express DMT1 and it will increase its transferrin receptor. So it seems that in the liver, a low oxygen state would actually cause the hepatocyte to pull in more iron, removing more iron from circulation. But at the same time, it will also express genes that will downregulate hepcidin production. So overall, although the liver may take in a little bit of extra iron, overall the iron status should increase. You also have to keep in mind that there's another sensor for intracellular iron. It's called the iron response protein and the iron response element. And whenever that gets upregulated, it actually turns off hypoxia-inducible factor. And I'll talk about how that works in just a second. Now, hypoxia-inducible factor doesn't just work in the liver and in the intestine. It also works in the erythroid cells and in the kidneys. And so when you have a gain-of-function mutation in hypoxia-inducible factor 2 alpha, it can cause polycythemia. This is simply interesting to note because it's only happened to 2 people that have been reported in the world. Most cases of genetic polycythemia are from a JAK-STAT mutation. So now we want to talk about iron excess. So you've had your iron coming in and it's been stored as ferritin. When that ferritin gets filled up, it gets full, you'll have labile iron starting to accumulate. Now I have to admit, I don't know if it accumulates in the Fe2 plus or the Fe3 plus state, but most of the experiments done to test its interaction with the iron response protein do so with divalent ions. Okay, so what I want to say is that the labile iron, that free iron, can bind to a regulatory protein, so the iron response protein. Now that might not be true, but we're just going to assume it is now because that's the classic canonical teaching. So it binds to the iron response protein, and then that iron response protein will become unbound from mRNA. Now the way to think of it is like this. You have your DNA and you've transcribed it into mRNA. You want to make a protein, but your mRNA gets clamped by this iron response protein. So this protein just jumps on, clamps down on it, and prevents anything from happening. If there is enough iron present, it will come over here and it will cause this iron response protein to be removed off of the mRNA, and then it can be translated by the ribosome. So what is the iron response protein doing? It's binding to a region called the iron response element. So some sources will call IRP the iron response protein, others will call it the iron regulatory protein, and the mRNA sequence the iron response element. Nevertheless, I digress. This is a picture of the iron response element, the mRNA folded up into hairpins, taken from Wikipedia. When the mRNA is folded up like this, iron response proteins can bind to it. So what you need to know in general is that it can be at the 5' end of an mRNA, where it is inhibiting its translation, or it can be at the 3' end of the mRNA, where it is inhibiting its degradation. If it's at the 5' end and iron comes in and causes it to be released, then that iron will basically upregulate the production of that protein. On the other hand, if it's at the 3' end and the iron comes in and binds to it, then it will allow that protein to be degraded. Thus, iron will effectively cause it to be downregulated. So it's really easy to remember. 5 is higher than 3. So iron with 5, you go high. Iron with 3, you go low. I do have to tell you, though, that this is not the full story. This is just the classic teaching. Now also the classic, the canonical teaching is that iron will bind to the iron response protein. So there's actually 2 iron response proteins. There's an IRP1 and an IRP2. And iron will bind to the IRP1 and cause it to be released, or it can bind with another protein to the IRP2 and cause it to be degraded. Both of those things might actually happen, but there was a great study done in, I think it was 2010 or 2014, one or the other, and it showed that when iron comes over and binds to, it actually binds to the iron response element. It binds to the mRNA, and what it does is it causes the mRNA to change shape, and by changing shape, it allows the initiation factor to outcompete the iron response protein for that binding site. Now, there have been some very stringent kinetic studies done after that first initial study that have added some weight to that story. So it could be that both of those things are true, but if I had to just pick one, I'm saying that iron is binding to the mRNA. So iron bound to it, it caused the iron response protein to be dissociated. So then what genes are affected by the iron response protein and the iron response element? That's a good question. So you have ferritin heavy chain, you have ferritin light chain. Both of those two make up the ferritin molecule. You have transferrin receptor 1, aminolevulinic acid synthase 2, or ALAS2. Now, ALAS, you'll remember, is the first step in heme synthesis. ALAS2 is only expressed in erythroid precursor cells. Every other cell expresses ALAS1. So thus, in the presence of iron, erythroid precursor cells will upregulate the production of aminolevulinic acid. This has important implications for porphyria, because you have two kinds of porphyria. There is a erythrogenic porphyria, and there are hepatogenic porphyrias. So for erythrogenic porphyria, you don't want them to have excess iron, because if you do, it will drive the iron forward to those porphyrin precursors and cause damage. On the other hand, if you have hepatogenic porphyria, you give the person synthetic heme as the treatment, and that synthetic heme actually stops the hepatocytes from producing porphyrin. And the main treatments for hepatogenic porphyria are actually heme, which is called hemin, the synthetic form, and glucose. High levels of sugar slow down the liver's production of porphyrin. Now, other things that are driven by this response element include the transporter that brings the metal into the cell, into the enterocyte, ferroportin, which allows it to leave the enterocyte, and the hypoxia-inducible factor 2-alpha can be regulated with the iron. Now, I tried to put a lot of the common names that these things go by in the literature. So DMT1 in the medical books is going to be called DMT1. In the science papers, it's going to be called SLC 1182. So now you also need to know, does it bind to the 5' end or the 3' end? Or in other words, is excess iron going to upregulate or downregulate this protein production? Now I'll go ahead and write decrease right here on DMT1. Now the 3' in general, if it's a 3', it will decrease it, and that goes with the classical teaching. Transferrin receptor is weird. I'm not sure I completely understand how it's signaling, but in general, a lot of the papers I've read say that it increases TFR1. To be honest, I'm going to say that it doesn't make any sense for it to do that, and so I'm going to say don't take my word for it. Go and check the literature and see if you can find something different and put it in the comments. Now, I'll also point out that the iron response element on DMT1, it can be spliced out. So DMT1 has, whenever the mRNA is being produced, it can be alternatively spliced so that there is no iron response element. So it can be both controlled by iron and not controlled by iron. And I'm also under the impression that ferroportin may be the same way as well, but I didn't get a definitive answer on that. Okay, so the reason that we have some of these oddities is, if you'll notice, look at hypoxia-inducible factor. It has an iron response element at the 5' end and at the 3' end. Essentially, what does that even mean? It's basically a fine-tuning, in that you should know these iron response elements, they all have a different affinity for the iron response, the iron regulatory protein. So you can actually have different sequences that will bind to the iron response protein. We don't even know what all of them are. In fact, there was a really, really good paper published a while back in 2010 that showed an algorithm to be able to find more of these things. And I actually adapted this list from that paper. Now you should also know that the macrophage can play a role in recycling iron. So you have your enterocyte, which brings in new iron. You have your macrophage, which will recycle iron. Usually it does so by endocytosing red blood cells, and it'll take up its iron. But then how do these things get rid of their iron? As you can see, the ferritin in the enterocyte is completely full. And as you saw from just a moment ago, that excess iron will bind to the iron response element and allow for 5' transcription of ferroportin. So then it can be shot out of the cell and attached to transferrin. I have all of these drawn wrong, so these are all a mistake. It should say Fe2 because every time an iron goes through a transporter, it has to be in the divalent state. And what happens is it goes through that transporter and it gets converted on the outside either by hephaestin or by ceruloplasmin in a copper-dependent manner. It will get converted back into an Fe3 plus state. So what you need to know for sure, inside the cell is a sort of a sketchy picture, but what you need to know for sure is that every time it goes through a transporter, it's in a 2+ state. Every time it attaches to transferrin, it is in a 3+ state. You need to know that copper and ceruloplasmin have a role in converting it back to that 3+ state. So it gets secreted and it binds to the transferrin. Usually transferrin is 30 to 40% bound. 40 is even high. But what you can see here is I have 3 transferrin molecules, and each transferrin molecule can bind 2 irons in the Fe3+ state. Now this is fine, but allow me to explain this paradox, this problem that I have with this. So I am told by every resource that I've read so far that transferrin is equal to total iron binding capacity. Now, what I just showed you is that I think that total iron binding capacity should be equal to transferrin times 2. That actually does matter, and let me explain why. Because you know that the saturation, the transferrin saturation of iron, is what we use to screen for hemochromatosis. So that's the serum iron divided by the total iron binding capacity. Now, a lot of times, I should put Fe instead of I, Fe for serum iron. And a lot of times on the papers and stuff we're given, we are not given the total iron binding capacity, we're given the transferrin. Sometimes we're given TIBC, sometimes we're given transferrin. If you can't convert directly between these two, you need to be able to know that. It's a paradox for me, I don't know, and so I emailed lab tests online last night hoping to get an answer. But anyhow, you can see that we get 30% bound normally. If more gets sent out of the cell, there is usually enough transferrin to scavenge it up rather quickly. But when transferrin saturation exceeds a certain point, then the iron still gets secreted out. That iron will be bound to low molecular weight particles, such as, for example, citrate, but there are several others. Whenever it is bound to a low molecular weight particle, in the jargon that is used is called non-transferrin binding iron, or NTBI. Sometimes you can think of this as being free iron, but most of the literature is not going to call it free iron. The problem is that this free iron has increased oxidative capacity, so it can damage cells. Now I put this here because that is true both of the free iron in the serum and the plasma, and it's also true of the free iron in the cell. So if it's not bound up in ferritin, it is potentially dangerous. So by way of quick review, iron absorbed in the duodenum enters the enterocyte after being reduced by duodenal specific cytochrome B, Fe3 to Fe2. It goes into the DMT1 transporter. It's oxidized back into Fe3 plus maybe, but surely whenever it gets bound up into ferritin. And then it's either bound to ferritin or sent out with ferroportin. Hepcidin is the body's major regulator of ferroportin. The enterocyte regulation of iron is controlled by a couple of factors. Oxygen tension is important because it will cause hypoxia-inducible factor, and that will allow the transcription of DMT1 and ferroportin. It's also mediated by cellular iron levels, so iron combined to the iron regulatory protein or the mRNA, we're not sure, maybe both. Once the iron has bound whatever it's going to bind, it causes the iron response protein to release the iron regulatory element, and then that will either inhibit or allow for gene translation. And then there are also systemic signals, most importantly hepcidin for the enterocyte. The enterocyte and the macrophage can send iron into circulation. Now, the hepatocyte can do this as well. We're going to talk all about hepatocyte in a minute. The transferrin levels should be around normal of 30%. Like I said, transferrin has 2 iron binding sites. This is currently a paradox for me, so I'll get back to you. And importantly, you should know that when transferrin saturation increases, then iron will slowly begin to attach to other low molecular weight particles. Now here's the erythroid precursor, and the erythroid precursor is saturated with transferrin receptor 1. Now's a good time to point out to you that whenever transferrin is bound to 2 irons, it is called diferric transferrin or ferritransferrin. Whenever it is not bound to iron, it is called apotransferrin. For simplicity, you'll know when I say transferrin which one I'm talking about. I will not use weird words just for the sake of using weird words. The transferrin receptor can bind to iron-bound transferrin at nanomolar concentrations. It does not bind very well to non-iron-bound transferrin. Once it binds, it will endocytose the transferrin receptor in a clathrin-mediated process. And once it's in there, you've got to get the iron off of the transferrin. So a proton pump will bring the pH down to about 5.5. And that will change the conformation of the transferrin enough that it will release its Fe3+. And I'm pointing out here that this is definitely Fe3+, because it has to be Fe3+ to bind to transferrin, and it just got released off of transferrin. An iron reductase will then convert it into Fe2+. It has to be in the 2+ state to go through the transporter. And after the iron has been sent into the cell, at that point the vesicle will move back to the surface, and it will rejoin the surface and spit out the transferrin receptor, because at that point the pH will change back to the systemic pH, and it will no longer have an affinity for the receptor. In this way, transferrin gets recycled. One review article I read made it a really important point to say that heme production requires transferrin-bound iron. It cannot use non-transferrin-bound iron, or NTBI. So the transferrin receptors have brought in a lot of iron, and those iron levels have went up to the point where the iron is interacting with the iron response protein. Interaction with the iron response protein in the erythroid precursor cells increases the production of aminolevulinic acid synthase, or aminolevulinate synthase, and it also stabilizes the transferrin receptor 1 mRNA. That's the current gospel I got from 2 papers. I don't understand how that happens, but I'm just going to take its word. Now the point is, and I've read this from multiple sources, I'm pretty sure I even read this in Harrison's about 6 or 7 months ago, is that when the erythroid precursor brings in iron, that stimulates it to bring in even more iron. Now the mechanism that we're talking about here doesn't make sense to me, because the iron response element on the transferrin receptor is the 3' end. So let's go ahead and draw our our RNA, and we'll say that this is my 5' and this is my 3'. And if my response element is right here, at this point I am preventing that poly A tail from being chewed up. I'm stabilizing the mRNA by having the iron response protein attached. When I get high iron and I cause it to deattach, that would destabilize the mRNA. So I don't know that I quite believe the mechanism that I'm being given. Nevertheless, that's the current gospel. But more importantly, the presence of iron is directly being tied to heme synthesis. Then the red blood cells get old, they time to break down, and the macrophage endocytoses them and takes up all of their iron. It breaks down the cell, and it breaks the iron off of the heme, but it's got to store the iron, so it's going to have ferritin. Now's a good time to point out that ferritin is made up of light chain monomers and heavy chain monomers. Now, the light chains are able to compress the iron into nice tight little places. The heavy chain has an oxidase capacity, so it can turn Fe2 into Fe3. The exact mechanism by which iron gets into ferritin and then the mechanism where it gets back out is still not known. It's active research. What's important to know is that ferritin is made up of 24 monomers. It is not exactly 12 heavy chain and 12 light chain. Different ratios appear in different kinds of tissue. But in general, one big 24 piece of ferritin will hold on to 4,500 molecules of iron. And basically, I already said this, I forgot that I had this slide. So ferritin is made up of 24 monomer subunits. The exact ratio is different in each cell, but all of those subunits come together, and we call it ferritin. Heavy chain has ferroxidase activity that can convert the Fe2 into Fe3, and the light chain promotes nucleation. That's going to allow lots of iron to be packed close together. The high iron absorb causes the production of ferritin. Both the light chain and the heavy chain have iron response elements. But there is a temporal uncoupling of the production of ferritin from the iron response element. So you can think of it as iron response element gets things going, but then it's like you push the car and then the car keeps rolling for a little bit. This allows excess ferritin to be made. It will become an iron poor ferritin, and the macrophage will secrete this out. This is secreting out of ferritin may or may not be done mediated through a protein called SCARA5, SCARA5. I don't really know, and I don't think anyone knows. But in general, iron secretion from the macrophage is via ferroportin, and it is hepcidin responsive. So again, another thing that has its major control mechanism via hepcidin. Now we get to the hepatocyte. The hepatocyte is two really important things. First, it's the major production site for hepcidin, and it is the major storage site for non-transferrin bound iron. The things that control the hepatocyte are inflammation, iron status, erythropoietic activity, and oxygen tension. So if you give somebody a phlebotomy, for example, it will shut down their hepcidin production. And it does that independent of the loss of iron. Now you should know that hepcidin is a type 2 acute phase reactant. So you'll remember acute phase reactants, it's a positive acute phase reactant. So whenever inflammation is present, its production goes up. And that fact that it's mediated by IL-6 via JAK-STAT pathway. Now, hepcidin in general is not simply mediated via JAK-STAT. It is only mediated via JAK-STAT via IL-6. So in high iron levels with the liver, it produces a molecule called BMP6. The BMP6 stands for bone morphogenic protein. So as you can guess, it has a role in the production of bone and the maintenance of bone, but it also has a role in the maintenance of iron. So the iron level is sensed and BMP6 is produced. Then the hepatocyte secretes BMP6. And oh my goodness, is that a BMP receptor over there on that hepatocyte? Oh, autocrine signaling! So BMP will bind to the BMP receptor. That will activate SMAD pathway, which will cause the production of hepcidin. Now the BMP receptor can do that all on its own, but it works a lot better when it has a co-receptor called hemojuvelin. Now you might be able to guess it's called hemojuvelin because it causes juvenile hemochromatosis type 2. More specifically, it's juvenile hemochromatosis type 2A. So the liver responds to the intracellular iron status via BMP, but the extracellular iron status, if you look out here, we are at 100% transferrin saturation of iron. And would you look at that, we have a transferrin receptor type 1 and a transferrin receptor type 2. What is this? Transferrin receptor type 2 is expressed in the hepatocyte. And these transferrin receptors are linked to the high iron protein, the HFE. You might know this as the classical adult hemochromatosis type 1 protein. There's a mutation in this protein at C282Y homozygous that causes adult onset hemochromatosis. So you remember that the intracellular iron stores cause the production and excretion of BMP, the BMP receptor, and hemojuvelin activation to signal to SMAD. So when the intracellular levels are high and the extracellular levels are also high, HFE joins in on that parade and starts to signal down this pathway to increase the SMAD. There's also talk of maybe possibly signaling through a MAP kinase pathway. And there's even a lot of strong evidence for a third uncharacterized pathway. And then the major regulator of hepcidin production is actually none of those things that I've just mentioned. The major regulator is not HFE, it's not SMAD. The major regulator is erythropoietic status. If you are getting a phlebotomy, if you have hemolysis, if you're getting exogenous EPO, all of those things will decrease hepcidin. So did you get that? Everything I said so far, HFE, SMAD, BMP, all of these things are positive regulators of hepcidin. The EPO, the erythropoietic function, is a negative regulator. There used to be a lot of strong theories about that being mediated through GDF15 or TWIS, TWSG1. Those theories are most likely wrong. The most likely thing is that it's being mediated through erythroferrone. So if we're making red blood cells, we don't want hepcidin because hepcidin will stop the production, the intake of iron to make those red blood cells. On the other hand, if we have low oxygen, hypoxia-inducible factor 2 alpha will become activated. It's also saying, hey, I need more iron so I can make more red blood cells. So it's like the two things to negatively interact here. And what it does is it causes the translation of a metalloprotease, MT2. And this metalloprotease actually cuts out the hemojuvelin. And that prevents the hemojuvelin from upregulating hepcidin. Okay, if you made it this far, you're really interested. So I'll go ahead and give you a couple of brownie points and bonuses. Uptake into the cell. I gave you basic education on it, DMT1. It is also probably mediated through a couple other things called ZIP8 and ZIP14. You can also import heme directly. I didn't mention this, but there's a scavenger on the macrophages, CD163, and then there are these other molecules that the exact role of them in heme scavenging is not completely known. And you can take up ferritin. So ferritin gets released by the macrophages. It's iron poor ferritin, but it gets released. It has some iron in it, and you can uptake that through the SCARA5 and the TFR1 receptor together. Now, if you have mutations in these, a mutation in transferrin, for example, is atransferrinemia. It's an iron overload anemia. You can have mutations in the transferrin receptors. Transferrin 1, you get an immunodeficiency and a mild anemia. Transferrin 2, you get hemochromatosis type 3. You have the channels, the DMT channel that we talked about. It can present with an iron overload anemia. Now, the idea about this is your enterocytes do not need to take up that much of the iron. They only need to take up a little bit. So out of all of the iron you eat every day, you only need to absorb a little bit. So dysfunctional DMT1 still has an extreme amount of reserve capacity. But once that DMT1 is isolated all by itself inside of an endosome, and it's trying to take the transferrin iron and secrete it back out into the cell, it can't do it all. It just can't get it all done. And so the erythroid precursors end up becoming clogged up and not able to get access to the iron. That causes signaling for more erythropoiesis, which causes the iron to downregulate its hepcidin, and you absorb more iron. The function of the ZIP8 and the ZIP14, not really known if there's any mutations in humans that are clinically important. Knockouts in mice of ZIP14 protect against hemochromatosis to some degree. The livers look better and the pancreases go longer before getting into diabetes. With heme import, this FLVCR2 is a known mutation in a disease called Fowler syndrome. Now, FLVC, feline virus something or another, basically it may have a role in iron regulation. It's not really sure. Intracellular storage of iron, so iron, ferritin with the light chain, the heavy chain. There's also mitoferrin in the mitochondria. You'll know that the iron gets made into heme in the mitochondria of every cell, and that heme gets used in the electron transport chain, the cytochrome. And heme gets used in the liver, the hepatocytes, in the P450 system. The liver is the second most abundant spot for heme and for iron. And then there's that labile pool, which is the part that is active in the IRP, IRE iron response. Mutations in ferritin light chain can cause neurodegeneration, seizures, restless leg. So cellular secretion, this is of course ferroportin. It's the main thing. It's the real important thing. There's also these feline virus thing. I like to think of it as the flavor. So I write FLV, change the C into an O, I got flavor. But the one I just talked about a second ago, that was FLV, that was flavor 2. This one's flavor 1, and we kind of know basically the function of this one, and it is to move heme out of the mitochondria. And there's some suspected function that it allows the erythroid precursors to interact with macrophages to exchange fully developed heme. And of course, there is also a lot of secretion of ferritin, and that makes up the laboratory basis for our ferritin test. A defect in ferroportin causes hemochromatosis type 4. This presents with iron overload and mild anemia. And alternative splicing of the flavor 1 gene causes the diamond black fan anemia, a very rare kind of anemia. The genes involved in the reduction of iron, you'll remember we talked about the duodenal cytochrome B, iron reductase. There's also a STEAP, a 6-transmembrane epithelioid antigen of prostate. This is important for the reduction of iron, so keep that in mind. Also prions. Prions in the brain can allow for iron toxicity. Now there's a whole, reviews written about iron in the brain and the blood-brain barrier and how endothelial cells kind of act like enterocytes in protecting the brain from excess iron. And so I'll let you look into that yourself if you're interested. Mutations in the STEAP protein cause hypochromic anemia. The oxidation of the iron, so you remember that I said in the plasma, ceruloplasmin, and also in the astrocytes. Now in the intestine, placenta, heart, and pancreas, hephaestin is important, and then in the placenta as well is zyklopin. Now deficiency in ceruloplasmin, this is not Wilson's disease. You get a low ceruloplasmin in Wilson's disease. That is because copper is not being attached to the ceruloplasmin. But if you have a deficiency in the actual ceruloplasmin gene, this causes a lot of neurodefect, diabetes, increased hepatic iron, and retinal degeneration. All transfer of iron within the blood needs to be in the Fe3+ state, and so if you can't get it there, you get stuck in the liver. There are no reports of hephaestin mutations in humans at this time. Hey, thanks for watching all the way to the end. I don't believe anybody actually will, but if you did, give this a thumbs up. I tried to get everything completely up to date as far as all of the science literature and everything on the basic metabolism of iron, because it really is so scattered and there's no updated textbooks at this point in time in history. So I did my best. I know I failed, but I did my best. Also, there were a couple of really good review articles that got me most of the way. One of them was a review article in Nature. I think it was either 2016 or maybe even this year, 2017. The other one was a review article in the New England Journal of Medicine from 2012. And so that 2012 article got me, halfway, and then that the updated information from Nature got me, up to 90%, and then the rest of it was just scavenging from hundreds of papers.